But I'm going to move over to ProteoDA. So Dennis mentioned this yesterday, but this is our package that my team put together. When we're trying to do high throughput proteomic analyses, we just wanted to make sure we had our standardized scripts that all of my team could run. We could keep up with the parameters and just all of those different things. So we turned, I had a new staff for a little while who's now gone on to bigger and better things, but we were interested in how do you even build an R package? So that's kind of where this came from, is like, this is everything I wanted to do, but how I turn this into a package, I have no idea. So Tim came in and helped us take all the scripts that me and Charity wrote and turned it into an actual package. What the difference is when things go into a package, now all of the code that we're running has to go through a bunch of tests. There's a bunch of testing associated with it, and it makes the installation very, very easy. So you can just install the package, not worry about all the other dependencies and all the extra stuff. So it just makes it a lot better for end user purposes. So this is published in the Journal of Open Source Software. Shown here is a little short description paper about it, but it's freely available on my GitHub page. And so I wanted to show this figure because it will give the highlight of what this package does. So I designed this to take the database search results, and you're starting from that point forward. I didn't want to make this so strict where it had to be scaffold or it had to be Spectronaut, it had to be a certain search engine. It can be wherever your protein intensities are coming from, it doesn't matter. You're going to have 3 files that you need to start running this package. Once you have those files in the correct format and loaded into the very first DA list, the rest of the package should run pretty well. That very first function is probably where you're going to have most of your errors when you're starting. Then that's probably going to be because something in one of these files is a little off. So I'll kind of walk through the example. But you have 3 files. The protein intensity is just the files with all of the intensity values of the proteins. The one column in that file that is specific is a UniProt ID column. You have to have UniProt IDs in there. Now you can use whatever identifier you want, but the column label has to be UniProt ID. Protein annotation is any extra information about the protein. So if you have your FASTA headers, your descriptions, your gene symbols, any extra information that you have about the protein will go into this one. And then the metadata is all the information about your samples. So the rows in the metadata, which are defining each of the sample names, must match the columns of the protein data. So that's how you're matching your information, right? So I'll show this here in a minute.
I have one question.
Yep.
How you deal with the redundancy and multiple entries for the protein?
Yeah, you can't have any duplicates.
But you need to do a curation before to be sure to include it.
Yeah, I mean, sometimes
because UniProt is no warranty anyway that.
No. So those, if a lot of those are ones, so one way to handle it is just put _duplicate and flag it as a duplicate, and then you can keep it all in your data frame if you want to keep both. But you cannot be a duplicate, or R can't make it another row. So, and you don't want to use dashes. Dashes are bad. Having a number in front of your sample name is bad. We need letters. So there's a little R things that gotta happen.
One more question. Any particular FASTA format from the UniProt? It should be Swiss, KB, should be Trembler, that doesn't matter.
It doesn't matter.
Okay.
Yeah. You're just pulling the UniProt ID column. Yeah, because that's gonna set your row names and the data frame in R. Yep. So once it's in here, we're basically made this DA list very similar to a DEG list and EdgeR and all the other genomic packages. So we're kind of following suit with genomic trends. And once you have that, then we have everything in a box is a different function within the package. So we have filtering, you can filter samples out. So you have an outlier sample, you can use the filter sample to remove that sample. You can filter out an entire group if you want to, filter out proportions by annotation. So you want to remove keratins, you can filter those out. And then we've got normalization. So normalization is my other tool that we wrote, ProteoNorm, and that one's running 8 different normalization methods and trying to evaluate which one's going to be better for the data set. Then you use normalized data to select the method, and then we write our QC report, and then we go to the statistical. The statistical design is all off Lima. You add in your design, add in your contrast. The difference with the package is we made it easy where you can type the formula and you don't have to figure out R code to make Lima run crazy models. So I wanted something that I could easily interpret. So we made that simpler, and then it fits the model, and then it outputs all of your results into the regular tables and the interactive plots and tables. So all of that's included in here. And after this workshop, I do have an example R script that shows you how to install everything, how to get started, how to do it. So I can send this R script to everybody as well with notes in it. So you don't have to remember everything I'm about to say. The other thing I also did is if you go to YouTube and search for Arkansas Embree, I've started a enhancing data science playlist, and I'm going to start putting more channels, more videos there. And I do have a video on ProteoDA going through all of it. So it's already recorded. I don't ever want to hear myself again, but it's it is there. So in this example, I have a couple of extra install package things here that you actually should not need. This is where I had issues on an older Windows machine, and it needed some extra things that weren't pulling from the package. And it's mainly this part of the code. So if you're running into errors, you might, I can help you with it, but you shouldn't need it. I just didn't want to lose how I fix things. So to install the package, we're using DevTools. We install it through GitHub. It's all under Byram Lab at ProteoDA. And we want to install it with the dependencies. So that's going to try to install all these packages for you without you having to do individual ones. Build Vignette will build the tutorial in an HTML document for you. Sometimes if you have a problem here, you need to turn that to false, and that will, sometimes that just kind of bugs out a little bit. Not sure why. So once you have it installed the first time, you don't have to run this every time. You just need to get it installed on your computer. Then after that, you do have to run the library to call that package that you've installed and make sure that it's running. So I've already loaded in the library, and then running this will take you to a new page on Chrome, and you can get the tutorial. And so this tutorial will actually walk you through everything I'm going to kind of talk about. This is the important part, setting up your data, your annotation, your metadata. I kind of got a couple examples through here. So I'm not going to go through this. It's part of the package that kind of walks you through everything.
Yeah. Can I jump in here for just a second? I just want to claim that I sat through a couple of these talks where she went through all this, and I haven't done programming since I took a Pascal class in college. That just dates me. And she sat down one-on-one with me, and I have done this successfully several times using Spectronaut data and the Diane data. And so everything she says now makes so much more sense now that I've done it a few times. So if you're sitting there feeling like you're sort of being swirled around in this maelstrom of data stuff, so hang in there. And like I said, she's done a video on it, and I'll be happy to, if you have some data sitting in front of you and you get the package or you want to start that, I'd be happy to Zoom with you and
that's when it makes sense.
Yeah, because really you have to have the stuff in front of you and go, that's what she meant.
So Dennis, what I hear you saying is that you'll go to everybody's lab and set this up for them. Is that
Especially to Puerto Rico. Yeah, I like Idaho and
where's Justin? We need Hawaii.
That's right. But I get to pick the time of the year to go to each place. Sorry.
Yeah, so,
So,
go ahead.
what cost for this?
This is free.
Oh, really?
Yeah, but this is one we developed. Yeah, no cost. I hit you with the cost already. That's why we're going into this part of it and more the detail, because this is free.
Rick would say, there's no such thing as a free lunch. So,
I just ask that you acknowledge the paper.
there you go.
That really helps me a lot. So,
Absolutely.
yeah.
And the data from Proteomics discovered can also be sent to this program.
Interesting.
Yeah. So this is just this package is only proteomics data, but it
But
runs.
from Proteome Discover, I don't know if we've ever
Yeah.
tried that.
No, you can.
I'm
It
sure we could if
works.
you get a report
You
file.
just
right?
have to. Yeah, it's just. I'm about to show that. Actually, we're on that part now. Yeah.
So which system is better for this? Windows or?
It'll run both.
or-
Anything. Yeah.
Uh,
So
you-
I'm running on Mac right now, and I also have that on my Windows machine.
How
So
about the Unix system?
Linux. Yep. So Linux. Yeah. You just start losing your RStudio on Linux. So that way I tend to run it through Jupyter or something. I've got it on Jupyter too. I've got it on all systems.
Gotcha.
So
So it's two characters.
Conda, I really like Conda environment. So set up my Conda environment and that has everything and then just run it that way.
So this is just for DIA data analysis or just?
No, it's
in-depth?
any data, any proteomics data that's intensity based. So
And then in terms of a Spectrum alpha file, do you mean the SNE file from Spectrum, for example?
I export the proteins report out of Spectronaut and then pull out the MS2 quant columns.
Oh, I see,
Yeah.
I see.
So I have another function and this is the public version. I also have a private version where we add more functionality to fix data files from Spectronaut, Scaffold, TMT. So there's a few extra functions that we do depending on the specific pipeline, but this one is just easier to start with right here. So your input data would look like this, as long as you can get it into a system like this. The way the example is set up, my protein intensities and my protein annotation are all in the same file. So you have to have, it requires this UniProt _ID label. It has to be all underscore. It has to be this specific format for that word because it's pulling everything based on this column. That's going to set your row names for the package. So UniProt ID has to be there. All this other information is session numbers, molecular weight, whatever protein annotation information can be in there. And then these are the names of this. In this example are the MZML files, and that's going to be each of the samples. So in here, when I load it up, I've got it. This is what you want to pay attention to. Are your values NA or 0? So one problem with Spectronaut is they have NaN. You've got to replace the NaN. This package will take an NA or a 0, but not an NaN. So that's one thing to watch out for because we probably need to add that back into the package, but I haven't yet. So typically I like to just replace all the NAs with 0s when I'm starting, then I know exactly what I've got. I know what's loaded in, and now we have another filter or another function in the package that you can change it to NAs. But so this also, let me load in. So the way to do this, I got my cursor in this RStudio and just hit the Command Enter or Control Enter, and it takes it down here to the console. And this is where it actually runs the code. So you don't have to actually type this all in. That's why I like scripts. But this is running off a system file. And so the system file are the example files associated with the ProteoDA package we've installed. You're going to want to change these two to point to wherever you've saved your input files. And I've got another example that I'll send you that has that information. So, but the other file is the sample metadata. So now we have what I like to do in my data, in my sample meta file is label a column that says data column name, because that is the values that have to match your column names in this file. So it just reminds me that that one is the one that matches between the two different files. So now we can say that first sample pool is pool number 1. The group is pool. It's all from the same batch. The second sample is sample 1, and they labeled them with numbers. So I can't have a number in the file, so I just put an S in front of it to correct it. And then that's normal versus cancer. So this is another melanoma project. But this is the critical part, is making sure that all these things match with your input data. So if you got an error, it's probably going to be in one of those files somewhere. So that puts it all in. You can run the head command. That'll show you the top of the different files. It's kind of hard to see in this little screen, but so now the next thing that I'm doing that may not make much sense, I have an input data frame over here that now has all of the samples, but it also has these extra columns of the protein annotation data. I don't need those in this file. So I've got to split it. I need to split this in between my intensity data and my protein annotation, because that's going to match what the input of when we're looking at these 3 files. I started out with 1 file where those were combined, and I need to split these into 2 to go into here. So that's what I'm doing there. And it's just the basic R subset command. So I take my data frame, and then this is what you're wanting to pay attention to, is the number. So let's see if I can make this a little bigger. Hard to get this big and all there. So the reason I'm starting this at number 5 is because this is 1, 2, 3, 4. My sample starts at column 5. So beginning at column 5, going to the end of the table, we'll grab all of the rest of the columns for the intensity values. So it makes sense probably if I run this. And now I have an intensity data frame that just has only the samples in it. And if I were to click on this, it'll show you now I've lost my Uniprot IDs, right? I don't have the names here at all. So we got to fix that too. So the other part that we removed, we want to save as the annotation. Yeah, I know. This is why you'll have me.
I just want to ask a question. How many of you guys have done anything in RStudio before? Done?
A few.
Okay. For those of you that haven't, I mean, she's explaining all this, but what you literally do is you hit Control Enter at each line, and then you hope it doesn't come up with an error. And if it doesn't, you've done it right.
Yeah.
And you just keep doing that all the way through her script.
And then when you hit an error, you just email me.
And then when you took my punchline.
Sorry. I know where this is going. No, it's fine. Once you do it a couple times, so you start to get the hang of it. But this is where we're about to fix our row names. So our row names currently just look like numbers. We want those to match our sample metadata file that we created. So now we're going to do that. And if we run our row names again, now they actually have the sample names and stuff. So everything is now back together. We got all of our data. This is why this part is so much more difficult, because with my private version, I actually have functions where you can just load in like the Spectronaut output, and it does all this for us and creates it and then builds I can go straight to the DA list. But you can't do that when it's public and you have no idea where the data is coming from. So this makes it a little bit more generic, and wherever you do your database search, it doesn't matter. You just got to get it into the right format. So this is where DA list is our very first function of the package. It's going to tell it the data is the intensity data that we named, annotation, sample metadata. All of these other are placeholders for what's to come in the package. So the design's going to be our Lima design that we create, the eBase fit, the results, and then any tags that we're collecting along the way. So this is just going to create a S4 object in R. And now instead of having something that looks like this, where it's one table, it's got all of the tables combined. So you have your data table that's in here, the annotation is here, the metadata is here. If you get an error, it's going to be here because one of these tables don't match. So you got 17 samples here, it's got to have 17 in the metadata file. So that's typically going to be where your first error is. Now, when I'm doing this one, we have pooled samples that are in there, pools of all the samples. Don't necessarily want it in the analysis. So I can filter by samples, and this makes, instead of using raw name here again, I make a new name so that I still have my raw values there. If I make a mistake and I need to go back up, I don't have to rerun everything again. So filter samples, some filtering out, and this means it's kind of a weird R, but take this group and remove it out from the others. So this is the convert missing values. We started with zeros, and we want to make our zeros NAs because Lima will omit the NAs in the calculation. And so I'd rather not. The only time I'll leave a zero in is if it's a known knockout and they want to have a true value calculated on that. Sometimes I get asked to leave the zero in and calculate on it. So I like having options. So we converted our intensity values to NA. So this is another section that you kind of have to decide. Depending on how many replicates you have in a group, this is going to filter out proteins that have only one value in one sample. So I'm not very, I don't want to keep a protein that was only identified in one sample. It hurts the statistics. So what this is the minimum replicates. Say if I had 8 replicates, I want at least 4 of those replicates to have a value in one of my sample groups. And these are all customizable. So I kind of generally take a 2/3 rule. If you have 3 replicates, I want 2 of them to have a value. But that's all dependent on what you're doing. And then the grouping column is very important. So in our sample metadata, the first file that you put into here, I have a column named group. And that is the one that's telling us, what these samples belong to. If you change this and make it a capital G, you're going to have to change this and make it a capital G. It has to match. So those things have to match. So when I do that, it's going to tell you we filtered out 203 proteins, leaving over 8,000 left for the analysis. So this, when you start to look at your QC plots and your violins and you see a bunch of low level stuff, allows you to have that flexibility of how stringent you want to be or not stringent. So there's some flexibility here. And then we're going to write our normalization report, which is my ProteoNorm report. Again, you want to make sure you're hitting the grouping column. We're going to name the directory where it's going to put the report, name of the file, and be careful with this one. Overwrite is set to true. So if I were to run that again, it's going to overwrite my existing file, which is what this just told me. I just overwrote my data, which is fine.
I just want to say one thing about that that's a little bit dangerous is that she has it set to name all of the files exactly the same: results, contrast, metafile, all that kind of stuff. And so if you open up one set of results and you try to open up another one, Excel gets really angry at you because it doesn't want to open up 2 files with exactly the same names.
I normally put it with the project ID in front of that, but it's the example.
Yeah, exactly.
Yeah. So the report,
If you have that set to false, will it auto-populate a different name or will it?
it'll ask you.
Okay.
Yeah, and then you'll have, so typically I would set it to false just to be safe. This one I'm just done a lot. Especially because there's a lot of times when you've got maybe 6 different groups and you only need to compare 2, and then so you need to subset the data a lot and run it multiple different ways. That's when the naming gets important. So this was patient data FFPE tissue, so the plots look a little funny. And that's kind of on purpose because a lot of them look really clean and it's hard to tell what we're looking at. But this is the log2 transform data only, so these are not normalized. It's kind of your unnormalized control. Each dot represents a group. So one group is the cancer group, one's the normal group. The top 3 plots are all variance, how much variability is in the data set. And then the bottom one is the correlation, how tightly are your replicates. Because this is the replicates within the group, not across group. So you want correlation to be high, you want variability to be low. If you have a lot of variability, you're gonna see that in your PCA plots. And yeah, so, but biology is very variable. The log 2, this is just a zoomed up picture of this one. You want the distribution of your changes or your fold changes to be centered around 0. Otherwise, the normalization method can kind of shift things negative or positive. So I'm looking for anything that's a high correlation and low variance. Typically cyclic low S or VSN. If it's low abundant, things like phospho studies, VSN tends to perform a little better. If there's everything is pretty similar, I just default to cyclic low S as part of the same lemma package. We did also add in MA plots just to see how the line is going to fit your data based on the different methods. So this is cyclic low S. You want that orange line to be fitting the data points centered on 0. You want it flattened across 0. Some of them kind of shift up or shift down, but this is all about the regression model that's happening in the normalization. So I won't bore you too much with that. I'm about to make a video. So that's the normal. So now that we look at it, we see the plot. These are the options in the function to be able to do the normalization, log 2, median, mean. We cut cyclic low S down to just cyclic low S because we're lazy. And so we're going to choose that one. And then we're going to write our QC report. And again, you want to watch kind of the overwrite method and all these things. And I'm going to make this false because I do have it already. See what happens. Yep. So I don't have a unique file name, so I didn't run because I already have it. Ugh. I really don't like that coming up. So so these are going to be your violin plots, just kind of looking at overall distribution. This is where I kind of pay attention to the bottom tails if we need to do more filtering of the proteins that have zeros or not. There's different things you can evaluate. Depends on the question. Here you got 2 outlier samples in this data set. You can see at the PCA level. You can also see at the cluster dendrogram level. So you probably want to go back and remove those 2 samples. So you can actually at that point go back to the filtered samples and remove things before you go forward. So it's kind of a.
Is there a statistical test that's defining those as outliers, or it's more visual?
It's more of a visual, and usually at this point, I don't remove them unless in a consultation, there's a reason that they need to be moved, because you're only looking at 26% separation here. It's 1 and 2 component. I don't get overly concerned about PCA plot, but some of my end users do. So you actually, having more data points in your stats makes your stats stronger. You're losing power when you move that out.
So you mean before you're loading your data and then you can add the filter. The data is filtered, right?
Right. Yeah. So you want to evaluate. So that's why it's not like when I run a project, we can just hit run and it's over in this corner and then I send out results to end users, because you got to kind of evaluate these different sets. And it depends on how they set up their experimental design, how that's going to play a role here. I got an example of a bad data set and a good data set. I can show.
Thanks.
But yeah, we may be kind of running out of time. Let's see here. So I like the missing value plot too. This one's in here. And this also kind of helps me to evaluate, do I need to filter more proteins with zeros or not? So this is only plotting the proteins that have a 0. It's not all proteins in the entire data set. Otherwise it gets kind of messy. But sometimes you can see a trend in the clustering of the samples. So those 2 outlier samples may have more zeros in them, and that's why it's the outlier sample, just because it's got those missing values at the protein level. And this will happen like serum, plasma samples for sure. If you have a lot of albumin, hemoglobin, that might be driving some of it, so you don't have as much of the lower abundant ones found in that sample. This is kind of where you can start to evaluate those questions. Okay, so we did our Q. So now everything's QC. So this is where we changed lemma design. So we're using our add design function. We have the design formula. We are running this particular formula right here. There's 2 different ways to run it. we don't have to have this tilde 0. You can just put group. If you do that, you don't get to define the way Lima is going to run your comparisons. So R really likes alphanumeric order. And if you don't set the levels of your factors, it will determine what is the reference, what is the control sample based on the alphabet. So I like to set my own direction of what the contrasts are going to be. So I use the no intercept model, this tilde 0 plus my grouping variable as the formula for the model. And then that way, when I set up my comparisons, I get to tell it the order that I want to calculate my fold change. And I know very specifically how it was done. And I also, this is just by convention that Big Omics uses the same format. Cancer versus normal, and this, anything that's a positive fold change would be up in the cancer compared to the normal control. And so this is going to determine your fold change directionality. And I like to name things this way. So at the end of the day, when I'm sending somebody results and I need to remember how the fold change was calculated, it's in the name. The name tells me the order. I guess I will not remember this later. So that actually ends up into the result file with that specific, whatever's labeled here is going to label on your plots and it's going to label on your tables that it writes at the end. So some people have really long phrases for their groups. Can't have a long phrase. I need this to be short. So we're gonna
20 characters or less. I found out the hard way.
Yes, that would be great. And then we're going to fit our model. This you don't really have to change. It's just as long as it is whatever that was. And then we're going to write our results. The results, so for the volcano plots where you see the coloring of the red and the blue, these are the thresholds that are set for that boundary. So if you want to lower your, instead of 2-fold change, a 1.5-fold change, you would lower the threshold here, and then you can regenerate the plots. All right, which one did I just run? Right, results. So once we have the results, then we just write out the tables, and I already have this. I'm not going to run it, and we write the plots. And so let's go see what that looks like. And then I know everyone's going to be ready for a break. Actually, let's see. I have it over. There's one. Yeah, so I don't know if I can make that larger. Not really. Anyway, you get a lot of files and you can't read it, but this is the cancer versus normal report, HTML. This is going to be the interactive plot that you just open in Chrome. And so it starts, you have plot type options. I think I can't make that one bigger. Yeah. So it started out with the adjusted p-value. You can change it to the raw value. That's going to change your axis. And we also have the MD plot instead of the volcano plot. So you've got both ways of looking at it. The MD, the red and blue, is based on the p-values. Well, yeah. And you have the average intensities and log fold change. So it's all there. But then you click on the little dot, hover over it, gives you some information about the protein. And then this is what I like to see is the individual samples within this group. So this is the normal group compared to the control. When you have a lot of spread of variability like that is where your stats are gonna suffer. I'll go back to the volcano. So a lot of times these down here on the end will just have a little bit more spread of variability of the points. And this, for this data set, it's totally expected. It's patient formalin fixed tissues. So they're a lot more variable than other ones we get. But you have the full data table is down here, all the proteins. So if there's one of particular interest, I'm just gonna make one up. You can click on it and then it highlights it where it is in the volcano plot for you. So this is really where the biologists like to play with the data instead of looking at an Excel spreadsheet. And they can search through here and look at stuff. And at the bottom, I do have the total number of proteins that are included in the data set because that kind of helps when I forget. So I could clear that out. But they do get The other one that I like that most people don't look at is the p-value histogram. So I've got some slides too on this. When you have a p-value histogram that looks like this based on the raw p-values, you know you're not going to pass adjusted. And this is because this is telling me that there's confounding factors in this data set, that these samples are not just cancer or normal. There's something else that's going on with the data set itself. There's a really handy, see if I can find where I put it. It's in the other. We found one of these statistical interpretation files from somebody else's blog. It was super helpful. But yeah, you get something like this where it's low p-values on this end, then basically this is just saying there's some other confounding batch in your study that you need to take account for and add that into the model. So instead of just group, I probably need to add group plus treatment or whatever it was that was the batching going on in this sample set. So it's not surprising that you don't have anything passing significance. So this is kind of one of those, and I'm not going to bore everybody with this, but I do have a bunch of slides on the details of lemma assumptions and models and how they fit and all this. So again, I'm about to make a video about all this. How do you go into confounding? And that just means that control group is also all female, or they're different things and you can't separate them by the group. Batching effects, how we set up batching. There's a good paper on proteomics on randomization and block randomization. How do we run things so that we're not just seeing a degradation of values over time, but we can still get the same values out of it. So we don't have time to go through all this today, but these are the models that I've been discussing, this means model and means reference. So I'm using the means model, difference between the groups. I'll send this to everybody. So any other questions real fast about the R package?
You have the ability to control, like, so confounding factor would be sex or age. Do you have the
Yeah.
ability, especially for larger data sets, to control
Yeah.
for age, for sex?
Yes, so if you have age in like a bin category, you can, sex for sure. So the way if I get back to my model here. So right here we have the 0+ group. This is where I put my my sex condition, just like that. If it's paired samples, there's a little extra thing that you have to do. The good thing, if you put a question mark down here and the function, we had to write the man pages. And so that'll also give you some examples over here of how to run things. So, yeah. But I try to add in kind of different options for the different things. Intercepts, no intercepts, additive models. So if there's an interaction or not, that's kind of the 2-factor design. This is the paired sample stuff. So there's a few extra. It hurts my head.
How about temporal samples if you have lots of time points?
I've just added that as another factor right now. There are other tools and I'm blanking on the name. There's another package for time data. It's in my notes. I try to avoid time data.
In addition to this is the word, are you also going to make a GUI word?
No, not on this, because it's all through RStudio anyway. Yeah, I don't have the capability of managing it is the problem. So like when we made a shiny and stuff, you have to maintain those and build it. It's a lot more work and I just don't have the manpower. So, yeah.
Are you familiar with the process?
Yeah. Yeah.
This one looks so better.
Yeah, there's another one too, Pro LFQ, I think that's our group. But we added a little bit more here. Yeah.
So this the input is basically just a test file. For the name of
Yeah. Yeah, I try to make the upload as generic as we can, but simple. Yeah.
I probably should know the answer to this and I don't. So when you do the phosphorylation analysis, I mean, since you're doing the same thing at the peptide level,
Yep.
is it the same?
Same process.
Okay.
Yeah. You just add the peptides instead of the proteins.
Okay.
And so that's why we run that one twice too. Yeah, so it's actually two separate analyses.
I think this is really good, but have you ever tried a different R packaging? I've been using the R packaging called ArtMS, AITMS.
I haven't, I'm not as familiar with it, but I know which one you're talking about. Yeah.
Yes, pretty much the similar, but the
Yeah.
packaging provide all the possible normalization and QC control and also data analysis files. So
Yeah.
looks very similar. Yeah.
Yeah, and we kind of built this one to run our high throughput stuff. So I've got more flexibility in my private version because we're testing other methods, new things. And so there'll probably be a 2.0 version come out when we add some more capabilities to it. But
Anyway, thank you very much. I'll definitely
But
try. Yeah.
One more quick question for the phosphopeptide level, it's not uniprot for the rows. So what's?
no, it's by, so there's a uniprot peptide key. So we put the amino acid and the number of the amino acid as the uniprot ID. So it's kind of
Gotcha.
merged together.
Okay.
And then that way too, because you got those two data files, protein groups and the phospho files where they come from. They have, it changes in one file. If it's protein groups, the ID column is the protein ID.
Yeah.
Phospho ID is the phospho ID, but those two
So
things link back.
You've appended them as a unique identifier. Okay.
Yeah, but there's if you're doing like histone PTMs, before I forget, there's actually another tool that we did for PTMs, PTM Viz, which is going to give you your bar charts across the histone and those histone mods. So that was done by one of my master students.
So one more quick question. In terms of this p-value in the volcano plot, so what's your suggestion? Do we use the raw p-value or you have to use the adjusted one?
It's going to depend on the biology. A lot of
Biology.
times your assumption is that most of the proteins don't change. So you shouldn't have a lot that's going on, especially if it's like patient stuff, you don't know, they might have more issues. So I don't mind going down to the unadjusted p-value, especially if you're trying to do functional analysis and pathway stuff. With pathways, you got to have 100 to 1,000 molecules is kind of the sweet spot. And a lot of times we have maybe 10 that are changing. You can't do pathways. So it really depends on the biological question. And so that's why we try to always have consults about the design and what the expectations are. If you have a really low effect size, I got to have more replicates. I can't do it with 5.
Yeah.
I just,
So
some of the diet studies, 5 is not going to show you anything.
so what's this default adjusting method using the
The
permutation?
FDR.
FDR.
Yeah, and you can change that in this setting too. So Lima's got all the different options, but that's going to be right here. Your adjusted method, the BH.
And mini hotspot.
Yep. Yep. So we try to make as many flexible points as you can, but you do have to kind of understand where your data is coming from. What are we analyzing? What's our goal? Do I want to just get the top hits? Then I might want to filter more of the zeros out. Do I want to do true discovery and leave everything there? Leave it all there.
Yeah, I think, and then we may talk about this a little bit more when we talk about, managing a core facility. But I think what we come up against is we want this done right. And so we do it ourselves. But the problem is, as we're going to have this conversation, is you do all this for the PI, and then they come back and say, "Ooh, can you do this contrast instead?
Yep.
Can you remove those samples and do it again?" And so then you're like, wow, maybe we should have something where then we present that to them and then they can do those sort of changes, which I think ultimately is what the biologist wants. They want to be able to have
Yeah.
control over that and be able to do that in some way. And so
Without me having to develop a software and a GUI like Big Omics.
exactly,
Otherwise, I'm like, use Big Omics.
right.
So the same problem is when you are doing pathways and gene ontology, because people are working in cancer in pancreas or whatever, and
Yeah.
you don't know everything, and they need to figure out what pathway are more interesting depending on what they were reading or something else. And this is,
Yeah.
they are annoying, but it's what it is.
Right.
You can make a big mistake doing something, we can
Yeah.
help,
Right.
but they need to tell us what probably they wanted to.
Exactly. So I try to put a lot at this point onto the end user. It's like, yeah, this is your data, your experiment. I can try to provide tools, and that's what I tend to do in post consults is point them to different free tools that they can use. There's EnrichR, there's a few, StreamDB things, but are we gonna take a break now?